Hey again, everybody. How we doing out there? Welcome back. We are going to be continuing our voyage today, starting Unit 5 and learning about muscles. So today we're going to be starting with some of the foundational characteristics and functions of muscles, and that's going to be really important as we move forward over the next 3 lectures. So here's some beefy people. You may have seen some of these individuals in competitions, especially if you're really into bodybuilding. But from our labs, you've learned a lot about these different muscles. You've learned a lot about how they function, where are they connecting, what are their actions, what are the antagonistic muscles, what are the synergist muscles. Hopefully those terms are familiar to you. And these are obviously some professional athletes who spend a lot of time developing these muscles that you learned about, right? Trying to focus on individual muscles, build them, getting ripped, getting swole, whatever you want to call it these days, but really working on that definition of those muscles. So they have a pretty good understanding of those different muscles in their bodies. Now, if you remember from labs, we have about 600 different skeletal muscles that are about half of our body weight, which is pretty incredible when you think about that. We've talked about 3 different kinds of muscles this semester: skeletal, cardiac, and smooth. And for Unit 5, we're going to be focusing mostly on skeletal muscle. Okay, we might talk a little bit about cardiac and smooth here, but you'll learn a lot more about those types of muscles in 202. So our major purpose is converting our ATP into some kind of mechanical energy. So taking our chemical energy and converting to mechanical energy, and we're going to see how this actually happens. So a quick review. So we have our 3 types of muscle tissue, and we've got our skeletal muscle over here on the left. And let's pull up one of these laser pointers. And so here we are looking at our skeletal muscle. Remember, our skeletal muscle is going to attach to bones and skin. So during your 3 labs, you learned about all these different attachment points and how those muscles, when they contract, what kind of movements are they responsible for. Remember, our muscle fibers are muscle cells, so we can call them kind of the same thing. And they are developed from myoblasts through a process called myogenesis. So we're not going to go into the details of that. It's just kind of want to throw that out there your way. And what we'll see with our skeletal muscle here is they are multinucleated, as you can see down in here. And they also have lots of mitochondria because, again, they need to create lots of ATP. Skeletal muscle here has these striations. It's voluntary and, again, really powerful for us to crank out weights and crank out reps, stacking some plates, if you will. Our cardiac muscle we find only in our heart. It's striated just like that skeletal muscle is, but it's involuntary and it's also branched. And so that, again, is helping that spread that electrical transmission across those cells. And then in our smooth muscle tissue, we find these in organs. It's involuntary and it's not striated. And again, it's sort of these really small fusiform shaped cells. Okay, good. So what does this tissue do for us? Well, lots of different functions. It helps us move around from place to place, as you can see these runners. Helps us with breathing, right? You learned about some different muscles involved with our respiration, so our intercostal muscles and our diaphragm, to name a couple. It helps with circulation, so even as we're breathing, taking a breath, it's helping move blood through our veins. It helps us with feeding and digestion, going to the bathroom and childbirth, and we talked about childbirth back in Unit 1 with that positive feedback loop. It helps us talk, helps us write, helps us with different facial expressions, as you can see with Jim Carrey up here in the corner, getting kind of crazy with his face. Helps us lift tires, right? So this is some really diverse tissue in our bodies. It is involved in stability, so we can see this amazing acrobat over here doing some crazy stuff on this little post here. We've got this athlete up here holding this posture on the rings. So not only is it doing that, but it helps keep us upright while we're standing. Okay, and so you can see some different types of posture here. Obviously, this is the best. It's going to tighten up those muscles, and so the more you work out those postural muscles or your core muscles, it's going to kind of help you hold your body in an upright position a little bit better. And for those yoga aficionados out there, are probably pretty familiar with these different poses and the muscles that are involved in that. What else does it do? Well, you learned about some sphincter muscles in labs, our orbicularis oculi and oris, so the sphincter muscles around our eyes and our mouth. But we also have a lot of sphincter muscles along our digestive tract that are going to kind of help control the movement of food and bile and blood. And so they are that circular muscle that's going to kind of open and close on these passageways. It's also involved in heat production, so you can see these thermal images of our body heat coming off of there. So about as much as 85% of our body heat is coming from these skeletal muscles. If you remember way back to Unit 1, and we talked about thermoregulation, as we get sensation coming in, pass down our afferent pathway to our integration centers, we get some kind of output. And if we're really cold, then we start to have some shivering, and that's going to help create some of this body heat and help warm us up a little bit. So what we're going to do is really integrate and tie together some of this bone information that we learned about in Unit 2. We'll talk about some of the histology that we also learned about in Unit 2, so with our tendons. We're going to look at what this muscle actually is, and as you can see, the kind of organization all the way down to this really tiny level. Hopefully you're looking at this and thinking, hey, I remember the slide of organization of our spinal nerves and how we organize that. You're going to see a lot of similarities with that. And then we're going to be talking about how our nervous system is interacting with our muscular system. So you can see up here a cross section through that spinal cord. We've got this lower motor neuron coming out of that anterior horn, running out through our spinal nerve. And here's that little axon popping out, again, not to scale here, and those little axonic terminals here, synaptic terminals, that are going to kind of communicate with our muscles here. All right, so that's kind of where we're going with this unit, and we're going to find out how all of that control that we talked about coming through those different pathways, like the corticospinal pathway, how that information is then connected to our muscle fibers and how that stimulates the contraction of those muscle fibers. Okay, so that's kind of where we're going with this unit. Now, what I wanted to point out with this slide is that our neurons are stimulating our skeletal muscle fibers to contract, but also notice all of these blood vessels in here. Okay, we have to have a lot of oxygen and glucose delivered to these individual muscle fibers. So you can see these nerves branching down here all the way down to these individual muscle cells. As well as these capillaries that are coming down all the way to these individual muscle fibers. And so again, lots of use when you think about working as a construction worker or a professional athlete. You are using a lot of energy to move and contract those muscles, and so we need to have a really good blood flow to help those muscles function appropriately. So let's start here and let's start organizing this. So what I wanted to do is start large and kind of work our way down. In chapter 4, we kind of took this approach with starting with these large pathways, and then we got more detailed to kind of then tie in unit 3 information. So what we're going to do here is basically start looking at the larger structures, some that we're more familiar with. So we can see this femur up here, and we see this tendon that's attaching our muscle, our skeletal muscle, to our bone. And so you spent tons of time learning about these different features in lab and different muscle types. And if you remember, that tendon is made up of that dense regular connective tissue. So it's going to be really strong in one direction, so that when these muscle fibers contract, it's going to put some kind of force on this bone and allow for some kind of motion. We can see that here in our skeletal muscle. And these skeletal muscles are then surrounded by a structure called fascia. And so fascia is our dense regular connective tissue. So it's going to sort of bundle our muscle, and it's going to separate our muscles from other muscles, or it's going to separate our muscle groups from each other. And it also is separating these muscles from that subcutaneous tissue that's a little bit more superficial. So if you remember, we have our integumentary system, so our epidermis, our dermis, and then we have that subcutaneous fat, and we have that hypodermis, and below that is kind of where we're going to get into our muscles. And so if you were to dissect down, you would then come in contact with this fascia that's surrounding this muscle right here. Now, the epimysium is a little bit deeper to that, and it's dense irregular connective tissue that is also surrounding that muscle, but it's blending into the fascia, so it's kind of uniform, but it also has these little projections right down here between these fascicles, and we'll talk about that in a second. And that's going to form our other layer called the paramysium. So if we look at those fascicles, remember we had fascicles in our neuron where we had those bundles that were separated from each other by connective tissue. And so here we can see that our paramysium is surrounding those fascicles. And so this is, again, dense irregular connective tissue. Remember, it's coming in from that epimysium, and it's going to surround those fascicles. And within that paramysium, we see larger nerves, larger blood vessels. We see some stretch receptors that may send back reflexes, or they're going to send back information about our location in the external environment, right, back to our brain. So some of that proprioception that we talked about getting sent back to our brain so that we can integrate and figure out where we are, what does our posture look like. Next, we have then, if we look at one of those fascicles, we have bundles of muscle cells, okay? And so they are cylindrical, they're multinucleated, as we mentioned earlier, and lots of mitochondria that are embedded within those individual muscle cells or muscle fibers, okay? So a bunch of muscle fibers is our fascicle, a bunch of fascicles make up our skeletal muscle. All right. So last then is our endomysium, and our endomysium is surrounding that muscle fiber. And kind of like we saw with that endoneurium in the nerve, we've got our endomysium, and this is areolar connective tissue. Again, remember, areolar connective tissue has lots of space for capillaries and for nerve fibers. So those nerves, as they're coming in, they need to penetrate down in between all this connective tissue and innervate into these individual muscle fibers or muscle cells. Same with those capillaries. Okay, so they've got to get all the way down into this really small microscopic level. So here is just a cross-section that I think is pretty cool to look at. You may have seen some of this in lab when you were looking at skeletal muscle. So we can see this longitudinal section of one fiber or one cell, so really long. We've got those striations in there, and we're going to look a little bit more closely at those striations and how they work. We've got all these multiple nuclei that are surrounding those individual cells. If we look at a cross-section, we can see a fascicle here, which are those bundles of those individual muscle cells. Okay, And so we can see that muscle fiber here. We can see those nuclei that are dark stained. And then we can see that paramycium that's surrounding our fascicle around here, and that endomycium that's surrounding the outside of each of those muscle cells. Okay, so make sure you're practicing that organization as you're moving through. Again, these muscles, I believe you learned all of these muscles in lab, and so if we look at our cross section through our leg here, we can see that we've got our gastrocnemius, so we've got that lateral and medial head you learned about, the soleus right in here. But what you can notice in this image is, this is just, I wanted to highlight sort of that compartmentalization and that organization of our muscles. So each of these muscles is surrounded by that fascia. So we've got our gastrocnemius, or sorry, our soleus here that's surrounded by that, our gastrocnemius, which is surrounded by that fascia. So it's organizing these compartments and sort of keeping these muscles separated. Okay, so if you were to go into a cadaver lab and you were to dissect in here, you'd be able to separate out each of these muscles from each other. Now, a blast from the lab past. You remember these terms: origin, insertion, belly. So the origin, if you remember, is our bony attachment that is stationary during a contraction, where the insertion is the bony attachment that is mobile during some kind of a contraction. So that insertion is moving towards that origin. The belly then is that thick part that's in between our origin and insertion, sort of in between our tendons here. And so when we contract, that is going to get a little bit thicker. When we relax, it kind of returns to normal. Now, if we look at our fascicle arrangement, there's a lot of different orientations of these muscles that you'll see. And so our strength of our muscle comes from the orientation of those fascicles. And so you can see there's fusiform, there's parallel, there's triangular, multipennate, circular, which we've learned about, right? So this is really important when you're learning about muscle physiology and you're learning about the forces that these muscles are putting on the bones. And so when you go into exercise science and physical therapy, you're going to be learning about how all these forces are interacting. But basically, when we look at these different shapes or this different architecture of muscle fascicles, there's sort of a trade-off with the strength of the individual muscle fibers versus the amount of muscle fibers that we can kind of pack into one area. And so when you think about our line of force, or our axis, basically is running from that origin to insertion, and so the muscle fibers here in this fusiform shape muscle are running parallel, and so they all those muscle fibers kind of act as one big they all kind of work together in the same direction. Whereas if we're looking at a multipennate or a unipennate, right, we have a reduced force of those individual fibers, but we get more fibers that can get packed into this area. When we think about big muscles like this pectoralis major or our deltoid, if you remember, we have these really big origins, and all of those muscle fibers then kind of insert on one point. So you have lots of force coming into one location on your arm, which is going to give you a lot more strength in that from that muscle. Now, if we talk a little bit about innervation, or the nerves that are coming in to stimulate a muscle, we can kind of think about nerve damage, spinal cord damage, or brainstem damage, depending on what effects are going on with that muscle. So we had talked a little bit about this back in Unit 3 and 4, but again, depending on where our damage is, or what muscles we can't move, we can kind of learn a little bit about which spinal nerve is being affected, or what area in the spinal cord is being affected. Remember, the spinal nerves are coming off of that spinal cord. They're passing through that intervertebral foramen, and they're coming out through that anterior ramus. And again, they're going to innervate into those muscles. Now we have cranial nerves. Cranial nerves are coming from the base of the brain, and so you just started the brain lab last week, and so these cranial nerves are popping out through those foramina that you learned about in labs, and they are innervating muscles of the head and the neck. Okay, and so if you remember, they're numbered 1 through 12. So where those spinal nerves are coming into muscles below the neck, those cranial nerves are coming into those muscles of the head and the neck. Okay, so go ahead and pause your video, take a few moments, practice labeling this diagram. Think about what is the function of this muscular system as you're kind of working your way through it. What are the different types of muscle? What are their location, appearance, functionality? And then again, label this and think about how is that different type of tissue adding benefit to this muscle? What is this organization doing for our muscle here? All right, welcome back. I hope that you were able to spend some time going through that and looking back at your notes and the previous slides in the lecture and sort of reinforcing that information in your brain before we move on. Now, this is an animation I pulled off of the internet, and I think it's just a pretty fantastic animation. So what's going on in this image? There's a lot happening here. So up here, we can kind of see this general overview of what's going on with muscle. So as we're contracting our muscle, you can see that that belly is getting bigger. We've got a nice scale here to kind of help us understand this. But then when we look at those individual fibers, we're seeing that they're also shortening and getting a little bit thicker. And then if we look even smaller, we're going to be talking about this, and that's kind of where we're going down here, is with these things called sarcomeres. And we're going to see how these sarcomeres function in muscle contraction. Now, what we're going to be doing as we move forward for the rest of this lecture is talking mostly about this region. We're going to look at some of the characteristics in this region, and this is kind of what we're going to be spending our last handful of slides on. The next lecture will be talking about this neuromuscular junction here, where we've got this action potential coming down and firing and some kind of signal going on here. So if you take a few minutes to just watch this animation and think about what we've learned with Unit 3 and 4. And so we see this action potential coming down, it's spreading down into here. And then we've got, remember, those lysosomes that have those neurotransmitters. Those neurotransmitters are being released in that synapse. And then we see something spreading, so probably another action potential that's spreading over this plasma membrane, also called the sarcoplasm. And then from there we see all these little white dots being sprinkled around in here. It's like one of those like snowballs, right? That you have for winter, and you shake it up and the snow falls down. But those are doing something because we see a little bit of highlighting going on with these different muscle, or these little proteins down here. You can also see these little things moving as that signal reaches them. So we're going to be trying to figure out all of this. And again, we're talking at the 1 micrometer here, right? We're talking really small. So this is what we're going to be moving on with. So again, I would recommend taking really detailed notes, pausing this video as we move through, and revisiting this a lot. So we're really focusing on the tiny portions of our body. Unit 4, we were really big talking about these neural pathways in the nervous system. So now we're shrinking back down. All right, so let's talk a little bit more about muscle, right? So again, movement is really important for us. Without moving around, we can't evade danger. We can't get food. We can't do anything really, and we would just kind of be stuck. So our muscle cells, again, are converting that chemical energy. We're taking ATP and we're going to create some kind of force on our skeletal system so that we have some kind of mechanical energy. Now, we're going to be talking about the physiology of skeletal muscles, and that physiology is where, you know, all of this training, so those of you that want to be athletic trainers or physical therapists, like, this is where all this information is coming from. And so, you know, when your coaches, maybe if you've ever played organized sports, have been like, hey, it's time to warm up. And, you know, young kids are like, I don't need to do that, I can just run around. Well, we really are actually changing the physiology of your skeletal muscle, and you're helping it perform at a much higher efficiency than if you didn't warm up. So it is a pretty important part of working out and competing. So some characteristics of muscle is that it's responsive. It is excitable, just like we saw with neurons. It's excitable, and so it responds to chemical signals. It can stretch, and it has electrical changes that pass along its plasma membrane. It conducts, so just like we saw with those neurons conducting an electrical signal via an action potential, here we're going to see that same thing happening where we have some kind of electrical change that's going to stimulate that action potential to travel along that muscle fiber, or remember, muscle cell. It's contractile. It has contractility, okay, meaning it shortens when it is stimulated. And we didn't see that in our neurons, right? We didn't see our neurons getting any shorter. And that's one of the nice characteristics and advantages of muscle. It is extensible, meaning it can be stretched between contractions. And it is elastic, so it returns back to its original length after it is stretched. Okay, so if we look a little bit closer at our skeletal muscle, remember it's voluntary. We have conscious control over it. We also can have unconscious control with reflexes, but we can then control it, and it's striated in here, so we can see these really nice images of striations. There's their friendly endomysium separating those muscle fibers from each other, and those multiple nuclei. So basically that muscle fiber can be as long as 30 centimeters, so pretty long. And again, muscle cell and muscle fiber can be used synonymously here. Now, the connective tissue components, if you remember, we are having our tendons and our ligaments. So our tendons are attaching our muscle to bone, and so we have that collagenous fibers that are really strong in that one direction, and that meshes into that periosteum, that connective tissue of the bone. Collagen, remember, is a little bit extensible and elastic. It stretches a little bit under tension, and it kind of recoils when it's released, but it prevents overstretching, right? And that helps protect our muscles from ripping or getting hurt. It helps return that muscle back to its normal resting length, and it is important in our muscle efficiency and our output, okay? So our power output from that muscle. Now, what we're going to look at is we're going to zoom in even more, and we're going to be focused in on one muscle fiber. So right here, we've got our muscle fiber or our muscle cell, and so we need to learn a little bit about the characteristics of one individual cell. Remember, this could be 30 centimeters long. Now, if we start here, we've got our muscle fiber, right? So that's just what we mentioned. So we got this circle around this entire muscle fiber. Our next structure is called our sarcolemma, and this sarcolemma is our plasma membrane. And I actually think that I misspoke before and I said the sarcoplasm, so I apologize for that. But our sarcolemma is that plasma membrane here. Below that is the sarcoplasm, so basically the cytoplasm of our muscle fiber. Then you can see in purple here we've got our nuclei, so our multiple nuclei that are just below that, that sarcolemma. They're kind of pushed off to the side because we need to make lots of space for these structures, which we're going to be learning about. So we can push those nuclei out to the side. We don't really need them taking up the space in that inside of that cell. We want lots of space for these structures that we'll get to here in a second. And then we can see lots of mitochondria, but we do want lots of mitochondria embedded within all these structures, because remember, those mitochondria are going to be making lots and lots of ATP. Okay, then what we see on the sarcolemma are these little openings to that we call that are going to lead into a structure called our T-tubules, or our transverse tubules. And so there's sort of these infoldings of the plasma membrane or the sarcolemma that kind of pass through and down into the cell. And we've got our terminal cisternae, and so those terminal cisternae are part of this sarcoplasmic reticulum. And so we're going to learn a little bit more about how all of this kind of plays a role in firing these muscles. So there's what we call a triad. So this triad is made up of two terminal cisternae and then the T-tubules that are kind of innervating down or infolding down and into the cell. And then that sarcoplasmic reticulum is going to sort of form a network around these myofibrils, which we'll talk about here. So you can see this network of sarcoplasmic reticulum surrounding the entire myofibrils. Okay, and so we can see that embedded down in here. This is going to be where our muscle fibers store calcium. So and that calcium is going to be really important in our muscle contraction. So we've talked about the importance of calcium in our synapses and in our nervous system, and now we're going to see how calcium is involved in our muscle system. We also talked about the importance of calcium in our skeletal system, and we learned about how our body regulates blood calcium homeostasis. So utilizing those osteoblasts and osteoclasts, as well as those hormones, calcitonin and parathyroid hormone. So again, kind of building those connections. This is another reason why we talk about that information earlier in the semester, so that we have a better understanding of how it's going to work with Unit 5 or in our muscles. All right, now those myofibrils are these long bundles, and those long bundles are basically taking up most of the space here within our sarcoplasm. Okay, so you can see these big bundles, and within those myofibrils we have myofilaments. Okay, and so those myofilaments, we're going to learn about how these interact, and so these are going to be actin and myosin. Okay, and that's kind of where all the magic is happening. We will also be talking about this structure called a sarcomere, and that sarcomere is basically from the Z disc to another Z disc. We'll talk about what that means, and we can kind of see where these myofilaments are interacting in this sarcomere. Okay. Perfect. So moving on to another image, what I wanted to kind of show you is just a sort of a histological view of what these sarcomeres look like, okay, under a microscope. And so when we talk about those striations are coming out of those sarcomeres, or are a result of those sarcomeres. So we can see over here on the right, we've got individual myofibrils, okay, that are within that one muscle cell. And then we have these sarcomeres here that bridge that run from one Z disc to another Z disc. So that sarcomere, we can define it as the functional contractile unit of this muscle fiber, okay, or that muscle cell. And these muscles are shortening because these Z discs are moving closer together, and therefore the sarcomere is shortening. So as these sarcomeres shorten, our muscles shorten. So everything is kind of tied together here. So we've got another sarcomere over here. We have another sarcomere over here. So all of these millions of sarcomeres are going to shorten and pull those Z discs a little bit closer together. So if we look again more closely, let's start to identify some of this and define some of this. Okay, so we have our thick filament and we have our thin filament. And so these two filaments are going to be responsible for moving those Z discs closer together. Okay, so again, our Z disc is out here on the periphery, and our sarcomere runs in between those two Z discs. Now, you can see that this thin filament is connected to that Z disc, and in the middle here, we've got our thick filaments that are connected along this M line. Okay, and so the M line is sort of in the middle of what's called this H band, and in this H band, it's just thick filaments. We don't see any overlap. In the A band part here, it's darker staining, and this is where we see the overlap between the thin filaments and the thick filaments. Okay. And this, we can see these tiny little dots or these tiny little heads, and we're going to learn a little bit more about what those are going to be responsible for pulling this thin filament closer together. Okay. That I band is sort of the lighter staining band that we see. So kind of when we're looking back here, this is that lighter staining band here, where that A band is that darker staining region, and you can see that M line right in here. And so just to kind of bring that back in here, so we don't have lots of the thick and thin filaments overlapping. So here we're just kind of seeing those thinner strands here. We see our Z disc in there, and then we see this protein titin, and the titin protein acts as sort of this spring, and it kind of helps to passively or the passive elasticity of our muscle fibers. So as they're contracting, there's this elasticity that allows that sarcomere to kind of return to its resting length. Okay. Just another view to kind of point this out. If we see that myofibril, we can see these individual sarcomeres on this nice stained image here. And then within that sort of cartoon image here, you can kind of see where these things correlate. On a three-dimensional image, remember, it's easy to forget that this exists in 3 dimension. So this is happening along that entire myofibril. So we've got lots of thick and thin filaments that are overlapping, and we've got these Z discs that are connecting those thin filaments to each other. And so that's going to be moving closer together. Now a little bit more about those sarcomeres. So the sarcomere, again, we've mentioned this multiple times, is from Z disc to Z disc, and that's our functional contractile unit. Because these move closer together, that has this larger effect on our muscles, which allow our muscles to shorten as we contract those. Okay. Now what's important to note is that these thin and thick filaments here are not getting shorter. Okay, they don't change in length. What is changing is actually the overlap. And so if we have my hand, and let's say this ruler is our thin filament, it's not changing, it's just being drawn over our thick filaments. Okay, and so when we relax, it's going to move back. When we contract, it's moving over. So nothing is changing in length except the overall muscle and that actual sarcomere. Again, here's another representation just to kind of show you how, in a 2D representation, how we have these thin filaments overlying or surrounding, I should say, our thick filaments. And when it's contracted, they've just moved closer together. So that band, right, our H band here, in between, that space in between those thin filaments, disappears. Okay, it goes away because these thin filaments have moved closer together. If we look at it in a three-dimensional view, remember all those thin filaments are surrounding this thick filament, and those are going to then move closer. But again, the filaments themselves are not changing shape. The sarcomeres are changing their length. Now, let's look at what these thin and thick filaments are. So our first one is called myosin. So our thick filament is made up of a bunch of these myosin molecules. And so those myosin molecules have these these tails here and these heads, and they kind of look like golf clubs, okay? Now, when we take a bunch of these molecules and we combine them, we can see this arrangement of these myosin heads on this thick filament, okay? And so on the right half of our thick filament, so if we're looking at this, and I used this for a thin filament earlier, but let's pretend this is our thick filament. Our heads on one half of the filament are going to be pointing to one direction, where the heads on the other side of the filament will be pointing into the other direction. So here you can see these heads are all pointing to the right. If we had the left side of the filament over here, they'd all be pointing to the left. Okay. Now our thin myofilaments, or our thin myofilament, is called actin. And so our actin molecules here are two strands of these little actin subunits, and they each of those subunits has an active site, and that active site, this is really important to note, can bind to a head of myosin. So when we think about those little subunits, one active site will bind to this head, one active site will bind to this head and this head. So you can see here we've got all these subunits, each with an active site that can bind to one of those heads. Now, tropomyosin covers those active sites on our actin molecules. Okay, so this tropomyosin is covering about 6 or 7 active sites. And then we have this troponin complex, and so you can kind of think of this troponin complex as the lock, and our tropomyosin is the door, and the active site is our doorway that we want to go through. Okay, but right now the door is closed and it's locked, and we can't go through there. Now, on troponin, there is a little binding site for calcium, okay? And that calcium molecule will bind to troponin, which will then move as kind of our key that will open the lock and then open our door and expose our doorway. Now, I mentioned titin, which is sort of our elastic myofilament. It's very springy. It's on each side of the thick filament, kind of helps anchor it to the Z-discs. It's stabilizing that thick filament and kind of preventing overstretching. So titin's pretty important in this whole involvement here. So, but that's about as much as we're going to be going into it. Now, again, you see that overlap, and we see a little bit more detail here. So we see those actin filaments, we see that tropomyosin and troponin, and there's no binding going on right now because the doorway is closed and the door is locked. We see those heads, those myosin heads here. They're ready to bind, but they can't. But we see on the right side they're facing to the right, on the left side they're facing to the left. Now, we have some accessory proteins that are involved in this system. We are only going to look at one called dystrophin, and this is sort of our most clinically important one. But there are multiple other accessory proteins that have multiple different functions, and so we're not really going to go into that. But the most important thing to know about dystrophin here is that it's transferring forces from, as you can see down here, from these muscle contractions or from those sarcomere contractions out to our endomysium. Okay, and if you remember, that endomysium is surrounding that muscle fiber. So it's helping to transfer forces, which help transfer that force into the connective tissue of the endomysium, the paramysium, and then eventually to the tendon. And it also gives us information about the contraction itself, so that we're preventing muscle injury or damage to the muscle fiber. Genetic defects in dystrophin can lead to what's called muscular dystrophy, and basically with muscular dystrophy, those skeletal muscles degenerate and they get weak, and it ends up, we see a replacement with fat and scar tissue. So the skeletal muscles are not functioning like they normally should. So that kind of brings us to the end of our lecture here. And this is just what we went over, is the basic characteristics and structures that are going to be involved in this process. So our next lecture will be coming back and talking a little bit more about the functionality of that muscle and how it's contracting and how it's connected into our nervous system. So as always, here's some review and reinforce questions to leave this lecture. So practice these questions, and also practice labeling this diagram. And as you're labeling this diagram, think about the structures and the functions of each of these things that you're going through and how do they interact. Thanks a lot for tuning in, everybody. Have a wonderful afternoon.